So today we'll be continuing from where we left yesterday. Question number 41. Most of the plant disease resistance are gene products contain enzymatic activities, leucine, rich repeats, transcription, repression domains, G-box domains. I have no idea. IDK. Out of the several gibberellins identified in the plants, which one of the following is not bioactive? Okay, another I don't know. There are quite a few like plant physiology based questions. Nitrogenase, a complex metal containing enzymes, is involved in conversion of N2 to NS3. Which one of the following metals is not involved in the activity of nitrogenase? Okay, so we'll go for a big guess because I don't know the answer for this. So molybdenum, iron, vanadium, cobalt. I'll go for cobalt. Let me get a minus. Which one of the following agents cause relaxation of, what is this, mesangial cells? Who are mesangial cells? I don't know, but I'll go for norepinephrine. A patient comes to a hospital complaining of vomiting and diarrhea. The doctor suggested that the patient take glucose electrolyte solution orally. Okay, glucose and electrolyte, very nice. Which one of the following membrane proteins is likely to be involved in dehydrating patient? Okay, wow. So I'll say the options are cystic fibrosis transmembrane regulators, sodium glucose transporter protein 1, insulin receptor protein, sucrose isomaltase protein. Okay, so I'll go for this sodium because the electrolytes and glucose is here. This is why cystic fibrosis. Why insulin? Next, which one is required for vitamin B12 absorption in small intestine? Sodium co-transporter, hepatic hephaestin, cobalophyllin. I don't know from where this question has arisen either. Which one is the correct sequence of events that takes place during the phototransduction when light falls into the retina? Closure of Na plus channels, activation. The thing is, I don't know, but I'll read the options calmly. Okay, calm down, people. Closure of sodium channels, activation of transducin, decrease release of glutamate, decrease in intracellular CGPM, structural change in rhodopsin. Wow. Decrease release of glutamate, structural change in rhodopsin, activation of transducin, increase in intercellular CGMP, closure of Na plus channels, structural change in rhodopsin, activation of transducin, decrease in intercellular CGMP, closure of sodium channels, decrease in release of glutamate, decrease in intercellular CGPM, activation of transducin, decrease release in glutamate, structural rhodopsin. No, I don't know. The LOD score of 3 represents a recombination frequency that is, I don't know who LOD is, so another IDK. 3 times as likely to the hypothesis of no linkage, 30 times I don't know. Okay, I do. What is LOD? I must have even read it, but I don't remember it right now. What is LOD score of 3 represents recombination frequency? LOD, linkage of distance, length of distance, weird answers. Weird answers. It is 3. Score is 3. So what is 3 times the hypothesis on linkage? 100 times. 3 times as likely. That's the hypothesis of no linkage. Go for this. Assuming that A, B, C and D gene are not linked, the probability of the progeny being this from the cross between this and this will be, parents will be, okay. Let's see. So parents has capital AA and small aa. Very good. So this was 1/2 for this. Okay, capital BB and B and B. So sorry. Let us see for AA and AA. So all will be this. All. There is no probability for this. It is 1. Okay. For the second one is capital B, capital B, capital B, small b. So it's BB, BB, B small b, B small b. So it is 1/2. Okay. Now CC will give CC only, so it is 1 again. Now DD is similar to this and it gave this one. Okay. It is also 1/2. So my answer is 1/4. I don't know if there is a different kind of mathematics going on, then it will be different, but for me right now it is 1/4. The newborn baby of a mother having blood group of this AB positive and father having blood group of O negative got mixed with the other babies in the hospital. Oh. The baby with which of the following blood groups is expected to be opposite couple? A baby can be anything. What kind of question is this? Okay, let us see. C. A, B, okay, O, this, and this. So this is not the one and this is not the one. And this is again not the one. So, you? Nice question. I like it. Next question. Which one of the following DNA markers can be used to distinguish between homozygote and heterozygote? DNA marker that can distinguish between these two? I have no idea. I have been seeing this a lot, so I will mark this. But don't do this, people. Please don't do this. This is a very bad habit of mine. Which one of the following is a fungal disease of plant? Wow, fungal disease. I'll go for cucumber mosaic because I read somewhere a mosaic being a disease of plants, fungal disease. Okay, I have totally no idea, but I'll go for that. Which one of the following influenza A virus subtypes caused severe avian flu and was responsible for disease outbreak in Hong Kong? Wow, this is a good question. H1N1 virus, we'll go for that. In the below phylogenetic tree of the selected taxa, this is the taxa and the phylogenetic tree. So A from this unknown place, it has arisen, okay, and they depart from here, and then they part here, and A is becoming something, and then there is departure and diversion or whatever the word is. B and C is becoming here, so they are a part of group A. This when departed, D and E and F and G is not in the group, okay. X Group X. This group X is monophyletic and group Y is polyphyletic. I don't remember the meaning of this. What is paraphyletic? Monophyletic. Monophyletic, paraphyletic. I don't know. So, I don't know. There's no other word for it. I don't know. Which of the following describes as the identification feature of non-poisonous snake? Nice question, nice question. I'll go for flat tail, broad transverse scale, third superossum. Maybe this is a very wrong answer, but I don't want to know right now. Okay, a road construction is constructed through wet tropical forest, following which of the population species of forest butterfly declines? Which of the is not a road is constructed through a wet tropical forest, following which the population of a species of forest butterfly declines. Road is being constructed, butterflies population declines. Which of the following is not a possible explanation for road causing a decline in forest butterfly population? Okay, road facilitates immigration of gap-loving species which compete with the forest species. Road facilitates increased movement of individuals in the forest, okay, within the forest, which reduces the genetic diversity. Okay, I'll say yes to this. Road internally fragments the habitat and negatively affects important microhabitat conditions. Okay, it can happen. Road facilitates invasion by non-native plants that displaces native hosts and nectar. How will road destroys? No. Road facilitates immigration of gap-loving species. Okay, so I will go for this. Which one of the following statements regarding normal distribution is not correct? It is a normal distribution goes like this. Okay, it is symmetric around mean, it is symmetric around median, it is symmetric around mode. Okay, but it is not symmetric around variance. Why would it be symmetric around variance? Tropical regions may have more species diversity because of the following possible reasons, except it is saying that the tropical region may have more species diversity because of the possible regions, except tropical regions have had more time to diversify good under relatively stable climate. Okay, then temperate regions, good. Tropical regions have high spatial heterogeneity. What the fuck? Greater biological competition in the tropics lead to narrower niches. No. Lower predation intensifies. Okay, so we'll go for number 3 here, but I am not sure. Okay, which one of the following statements is correct? Ectomycorrhizal association predominantly produce phosphorus limitation, and endomycorrhizal association produce both nitrogen and phosphorus limitation. I don't know. Okay, I don't know. I'll learn. We can learn it. Which one of the following statement is correct for the following association? Allotropic speciation occurs between adjacent population. Parapatric speciation occurs between geological. Listen, allopatric are like this. Okay, parapatric are like this. Sympatric are like this. Sympatric speciation occurs when continuations are fragmented. Wow, what a difficult question. I'll go for third because this is the only one looking like the right answer. Next, which one of the following does not contribute to the microevolutionary change? Random meeting. According to the Hamilton's rule, who is Hamilton? Altruistic behavior can evolve when RV is greater than C. What is R? What is B? What is C? Where B is the extra benefit gained by the recipient as result of altruistic RV is greater than C. Benefit. C is the cost to the actor arising from performing the altruistic act, and R relatedness. I don't know between them. I don't know. Individual on the altruistic act and offspring. Okay, first relatedness and benefit is greater than the cost. Okay. Individual performing. Once again, what is the question? According to this. Okay. The altruistic act and R is a relatedness between the individual offspring and performing the individual performing the altruistic act and the offspring of the recipient. Individual performing the altruistic act and the recipient and the offspring of the individual performing recipient performing the altruistic act and the member of the population. Population individual performing the altruistic act and the offspring of the recipient. I would say go for this. Okay. Analysis of the nucleotide sequence of the ribosomal RNA are particularly useful in the evolutionary studies of the living organism because of the following reasons. Except RNA is evolutionary ancient. rRNA is evolutionary. Wait a second. Is particularly useful for the evolutionary studies of the living organisms. Why? Because of the following reasons. Except rRNA is ancient. Okay, I don't actually know about it. I'll just agree and not agree with what I feel about it. Okay, no free living organism lacks rRNA. Oh, true, that is true. Ribosomal rRNA, since critical for translation, can undergo lateral transfer amongst distant species. What? RNA has evolved slowly over geological time. Maybe. So I'll go for answer number 3. Which of the following is the most appropriate definition of gene pyramiding in plants? What is gene pyramiding? Introducing different gene of resistance to a specific pest in different genotype. Introducing a single gene for resistance to a particular pest in the different genotypes. I don't know basically. I don't like this. What is this? Ecology and evolution plus evolution mashed together. I don't know. To test the impact of CAMP on protein kinase, a conformation in cells, an investigator from FRET biosensor was fusing to fluorescence protein at the NNC terminus of protein kinase A. In absence of CAMP in the cellular milieu, no FRET signal was detected. However, upon CAMP addition, a strong emission of this was observed. Configuration of fluorescence but used to investigate was, okay, we'll read it once again. To test the CAMP protein on protein kinase by fusing fluorescent proteins, C-terminus and N-terminus kinase of these. In absence, no fluid signal was detected. However, camp additions, a stronger emission of this was observed. What is the best configuration of fluorescent? Oh, I don't know. Truly, I'd like to know. Techniques, huh? In bioremediation by microorganisms detailed below, choose the correct. I have not done, nobody, like for 5 years, not read about this. The organic contaminants provide a source of carbon dioxide. This is a new word for me. The bacteria do not get energy degrading contaminants. Bacteria can produce oxidize or reduce species that can cause metal to precipitate. Bacteria on contaminants by aerobic, which is incorrect. So, the organic contaminants provide, okay, the bacteria do not get energy by degrading contaminants. Bacteria can provide oxidize or reduce species that can metals to precipitate. I'll go for B. I don't know. Multimeric protein when run on SDS gel showed two bands of 20 kDa and 40 kDa. However, when the protein is run on native gel, it is showed that the single band of 120 kDa. The native form of protein would be. I have no idea. A solution contains NADH and NAD plus, both at this concentration. If NADH has molar exchange coefficient of this and that of NADH plus is negligible, the optical density observed when a conjugate of will be. Absorption is equal to this. B into CB is path length. Absorption, I have no idea if this is molarity. This is concentration. This is the only formula that I have read that has path length, okay, molar extension coefficient and concentration. Okay. But I don't know about optical density measured in a cuvette of a. I don't know about optical density. Maybe it is also talking about absorption but in a different manner. Once again, we'll read the question. A solution contain NaDH and NAD plus, both have 0.1 mm concentration. If NaDH is a molar coefficient of 6220 and at another plus is negligible, path length is 5 mm. So this is equals to this. This will go with this. This 10, 10, 30, 31. Okay. Now we have two options. I don't know right now about what the conversation must have been happening here and here. So it is this. This is millimolars. normally we take it in molars. Partner is in millimeters. So it goes like 1, 2, 3, and then 1, 2, 3. 1, 2, 3. Because this also does not have a unit, right? How would I know which one? So we'll go for something like 1, 2, 3, 3.1. There's no 3.1 here. 1, 2, 3. This. I have this. No. This. No. Oh dear Lord. I'll just go for fourth option. Fourth option. I don't know. Okay. The orientation of clone DNA fragment gene in a plasmid vector can be checked by. Orientation can be checked by PCR using two genetic gene specific primer. Restriction digestion with enzyme that has a single restriction within the chromosomal and maybe PCR using a combination of one gene digestion of an enzyme that has two restrictions. I'll go for the second one. What I feel like it. I have no special reasons for it. The emission maximum of tryptophan fluorescence in the protein is this 335 nm. Suggests that the tryptophan A, that tryptophan is hydro. How would I know? I don't know. Okay, so we'll continue for the group C part sometime else. Another day, maybe tomorrow. Okay, so have a good day, have a good night. I'm having a headache.